Hello, my name is Connie Liu. I'm an undergraduate at MIT, and today we're going to be doing a concept question about the first cells. So, the way we're going to format this is through a Venn diagram, as you can see on your screen. This is showing the comparison between what protobionts, or the first cells, were like compared to current cells, such as prokaryotes. I chose to compare it against prokaryotes because those are simpler than eukaryotes and would probably have more similarities and fewer differences with these protobionts. If you start looking into eukaryotes, they have a lot more organelles, they're really complex, and then things get a little too complicated to even compare them. So now let's look at the similarities between protobionts and prokaryotes. So some of the similarities arise from basic things that are needed in order to be considered alive or life. Both protobionts and prokaryotes can replicate. Both of them can also metabolize, and both of them have some sort of lipid membrane in order to contain all of the things inside of the cell, inside an enclosed space. So this membrane is really important to protect it from the outside and to keep everything contained together. Metabolizing is also important in order to be able to process everything that's going in and out of the cell. Then replication allows these cells to be able to sustain themselves by creating more of themselves whenever need be. So the reason that protobionts were able to beat out other molecules evolutionarily was because they were able to replicate. However, replication is a little more difficult when it comes to prokaryotes versus protobionts because protobionts in terms of instead of prokaryotes, because protobionts are using what are called ribozymes. So that's a little different than current cells. Ribozymes are essentially the RNA genetic information. So they're the RNA that are able to both self-replicate, so they don't need additional proteins to do that, and they're catalytic, so they're able to catalyze these reactions by themselves, so it doesn't take a very, very long time. They also have no ribosomes, whereas current prokaryotes do have ribosomes. The replication techniques used in prokaryotes is also much more similar to that we've discussed before, with a lot of proteins being involved, catalyzing the reaction, and this genetic information, instead of being stored as RNA, is stored as circular DNA. And then the original protobionts actually formed spontaneously from organic compounds, whereas we can't say that about prokaryotes because they had some ancestor, probably protobionts, where they arose from those other cells. So those are the main similarities and differences between the first cells and current cells today.